Hi, welcome back to 15 Minutes with Dr. Vaccine. Today we have 5 vaccine experts who are coming to give you all a lesson. Welcome Dr. Kate, Dr. Emma, Dr. Matthew, Dr. Samin,
Hello there.
and Dr. Suraj. The vaccine that we are going to talk about today is the most important vaccine of the last 60 years. It is called Polio Vaccine. A lot of you may be wondering how this disease was spread and how the vaccine can reduce the number of polio patients around the world dramatically. Our experts have the answer for you.
Polio or poliomyelitis is a disease caused by a virus which enters the body early. It most commonly affects the children, although it can occur in adults, which can be infectious but mainly symptomatic.
A low proportion of sufferers can develop serious symptoms including stiffness in the neck, back and legs. However, in some extreme cases it can cause paralysis. In the worst cases, the virus will paralyze the muscles used to breathe, leaving the victim unable to breathe on his or her own.
The disease has existed for thousands of years, but the virus was only conclusively identified in 1908. It has now developed to become a major childhood disease with children most vulnerable to the virus's effects.
It was predicted that total eradication of the disease would occur by 2018, but most recently there have been widespread outbreaks in Asia and Africa. There is no cure once infected.
This is why prevention is so important, especially regarding vaccine production and distribution. Optimizing the manufacturing procedure used to produce the vaccine and make them as accessible as possible is key to eradication.
Now let's start with Dr. K, who would explain to us antigen identification.
Hello, in this break, we are going to explain you how do we produce the polio vaccine. Let's start with the antigens that used to produce this vaccine. Polio or poliomyelitis is a disease caused by a poliovirus, which enters the body through a mouth. The disease occurs before first vaccine was used over 35 years. The vaccine that used to against the disease is called inactivated polio vaccine or IPV, which is extremely efficacious in preventing individuals against poliomyelitis. This vaccine is a mainly trivalent produced by using 3 wild-type poliovirus strains, which are poliovirus type 1, 2, and 3, or Mahoney, MEF1, and Suricate, respectively, and grow in a type of monkey kidney tissue or cell culture, or inverted cell line. After that, inactivated by formalin treatment. There are 2 distinct antigenic forms of poliovirus exist, which are C and D antigen that used into polio vaccine manufacturing. The C antigen is expressed on non-infection empty particles, and D antigen is expressed on native infectious virus. However, the native D antigen can be changed into non-infection C antigen by heating at 56 degrees Celsius. And high antigen content vaccines in general take form of highly purified preparation of the antigen with the minimal contamination by the C antigen.
Dr. Emma will explain to us the fermentation process.
The poliovirus from which the vaccine is manipulated needs to have the ability to multiply in order for the polio vaccine to be mass-produced. This can be done through a fermentation process of a host cell. Firstly, a cell culture is created in which tissue from the species selected as the host cell is added to a growth medium in a culture dish to allow the cells to grow. This method offers greater control than using live animals and allows for production in large quantities. Virus cells are chosen as the host cell as they are sensitive to infection with poliovirus. The virus cell line was the first continuous mammalian cell line found in African green monkeys and is now the most widely used cell line for not only polio vaccine fermentation but other viro-derived human vaccine production. Vero cells are used instead of kidney cells directly removed from the monkey as they can be easily banked. The high degree of characterization of these cells that can be obtained reduces variability in samples and contamination from other viruses. Monkeys are highly susceptible to simian viruses, which can easily become part of the final poliovirus vaccine from entrainment with the poliovirus. Poliovirus can also grow in human embryonic brain tissue. This type of tissue grows fast, giving it an advantage over using vero cells as the host cell. However, it carries the risk of damaging the nervous system of the receiver of the vaccine. Vero cells are preferred over the use of these cells since they are more easily adapted to and grow more effectively in industrial scale fermenters, producing a higher yield. A higher vaccine purity is also attainable as less contaminating species remain after fermentation. High production yields and purities lead to a more efficient and cost-effective production of vaccine. Vero cells require carbohydrates such as glucose or galactose and nitrogen derived from amino acids like serine or glutamine as nutrient sources. Serine is preferred over the use of glutamine as a waste metabolite ammonium accumulates as it decomposes. These nutrients are made available to the cells by the chosen growth medium. In industry, growth of vero cells occurs on microcarrier beads and large-scale fermenters. This process can often take up to a week as a lag time is generated while cells get used to the environment and attach to the microcarriers. After the initial lag, the cells begin to double in layers, resulting in an exponential growth rate. Growth may become limited due to lack of nutrients, surface area, and accumulation of waste metabolites. Conditions in the fermenter, such as temperature, pH, and oxygen levels, must be monitored, as deviation from optimal conditions can cause agglomeration of species within the cells, breakdown of proteins into shorter polypeptide chains and amino acids, and changes in impurity levels. The number of multiplication cycles in each batch also has a significant impact on downstream processes and the final quality of the product. After fermentation, the poliovirus enters the cells with the aid of a viral vector, where infection occurs.
Now that we understand the fermentation process, Dr. Samin will explain to us product formulation.
All vaccine formulations combine chemical substances, including the active pharmaceutical ingredient, the API, into a suitable dosage form. The ideal vaccine product must be water-soluble, chemically stable, and have good access to the site of action. Initially, pre-formulation needs to be carried out where the API's properties are fully assessed. Depending on the properties, the types of excipients can be determined, which also depends on the dosage form, for example, tablet or capsule. Inert dilutants or fillers are added to produce vaccine doses of a manageable size. The drug load, that is, the ratio of API to the total contents, needs to also be considered. Binders and disintegrating agents will need to be added to ensure the vaccine is consumed at a suitable rate in the gastrointestinal tract. Ultimately, the drug form depends on the route of administration. The polio vaccine is named Inactivated Poliovirus Vaccine, IPV. It has two main forms. These are the parenteral and oral administration forms. The vaccine consists of a sterile suspension containing three types of poliovirus: Mahoney, MFE1, and Salkert. The viruses are initially grown in monkey kidney cell culture with newborn calf serum to allow for sufficient growth. The concentration of the serum is later reduced for the final vaccine form. Two antibodies, neomycin and streptomycin, are added to the formulation along with formaldehyde, as known as formalin, which is added as a preservative to kill the live viruses. This results in an activated form of the vaccine. The main vaccine contents include calf serum protein, formaldehyde, i.e. formalin, monkey kidney tissue, neomycin, 2-phenoxyethanol, polymyxin B, and streptomycin. It's essential that extensive research and development is carried out at the formulation stages to ensure the cell culture used does not contain any other additional virus types. If this does occur, the end drug product can cause undesired consequences to human health. For example, an earlier type of polio vaccine produced contained an additional cancer-causing virus called SV40, which mutated healthy genes in humans. As a result, approximately 98 million Americans were exposed to this particular virus between 1955 and 1963, meaning they are all at risk of developing cancer.
Downstream processing is now explained by Dr. Matthew.
The downstream process generally involves the purification of the product and the disposal of waste materials. Polio vaccine production is no different, with the purification process split into 5 main sections, including clarification, concentration, size exclusion chromatography, ion exchange chromatography, and the inactivation of the vaccine. In this short section, these main steps, also the disposal of waste, will be briefly described and explained why it is a vital part for the quality, efficient vaccine production. Firstly, clarification. This involves purification via a normal flow or dead-end flow filtration device involving a millipore POD holder containing heterocyclic compounds, followed by an express SHC filter. This short video shows the basic mechanism for this process.
Dead-end filtrations consists of forcing a solution containing suspended solids directly through the membrane structure. Solids retained by the membrane collect on the surface of the membrane media, continually reducing the permeation.
The next step is concentration. A product form needs to be more concentrated by removing fluid whilst retaining solute molecules to be an effective vaccine. The concentration process of the vaccine is very similar to the clarification process, except using a tangential flow filtration with smaller filtration devices, sizing at just 100 kDa. Here is the basic mechanism.
Process fluid flows along the membrane surface rather than passing through the membrane structure. With fluid being pumped at a high velocity parallel to the membrane surface, tangential flow filtration prevents the usual pluggings caused by dead-end filtration. As a result, TFF is an efficient way to separate streams that would become.
There are 2 chromatography units involved in the purification process. Firstly, size exclusion and then ion exchange chromatography. In the polio vaccine size exclusion chromatography, a 20 mmol phosphate elution buffer is used and maintains a neutral pH of 7. Size exclusion chromatography, a form of chromatography that separates molecules by their size, which is typically defined by their molecular weight. As you can see in this clip, the larger molecules are absorbed first, separating the compounds respective to their size. Now for the ion exchange chromatography. For this chromatography unit, the same elution buffer for the size exclusion chromatography is used, and also uses a pre-packed column which may be cycled multiple times per batch and or used for multiple batches. Post-chromatography sterile filtration is required to prepare for the inactivation of the vaccine.
Separation in ion exchange chromatography depends upon the reversible adsorption of charged solute molecules to immobilized groups of opposite charge.
This clip shows the basic mechanism for this process. Once purified, the vaccine is inactivated. The purified virus is stabilized with concentrated M199 medium containing glycine. The standard inactivation method involves the addition and incubation of 0.025% formaldehyde for 3 days at 37 degrees. An intermediate filtration is performed at 6 to 8 days, and the resulting bulk is stored at 2 to 8 degrees. Finally, for the waste disposal. Disposable components being used in the vaccine production include systems for process fluid mixing, storage of bulk materials, and product storage, filtration and chromatography, as well as distribution manifolds, sensors, connectors, and production scale bioreactors. Decontamination and sterilization of these can be done by fumigation and must be performed on any substance leaving the plant site.
Dr. Suraj will now explain engineering challenges and biosafety.
The polio vaccine produced by Salk was very effective, but it required a large dose and had to be injected onto the arms of children. Improvements were needed, and the fact that this is a virus easily transmitted orally, it meant that the vaccine could also be developed the same way. Oral vaccination is more effective because it is a live virus. However, it also raises concerns as it could convert to a disease-causing form, whereas the injected version is not live, hence why higher dosage is required. This also makes it easier to distribute around the world, as an injection would require skilled medical personnel, whereas an oral vaccine could be self-administered. This is easier to take in certain remote populations. The World Health Organization knows which communities still have polio present. The surveillance required for this is an example of how engineering approaches are utilized in solving medical problems. If a large-scale immunization effort was made to eradicate polio from these communities, we may be able to eliminate polio the same way we did smallpox. Other engineering problems include producing and distributing sufficient quantities of the vaccine reliably and safely. Those efforts have failed due to the resources needed to do this. Also, governments are not stable in some places where the disease occurs, and there may be civil unrest, making it difficult to give vaccines. Some communities are suspicious of Western medicine and are not willing to try these modern methods. There are still several challenges to be overcome to complete and maintain global polio eradication. This includes biocontainment of wild polio viruses, for example those used in IPV production, to prevent it escaping into the environment from facilities. Further challenges include prevention and control of outbreaks from OPV strains which may have evolved into wild viruses. Immunization with IPV or OPV has reduced the risk of the disease spreading to the general public. This has been further reduced by biosafety practices that help prevent contamination of the environment, as reintroduction of the polio virus into the community from the lab will present a danger to polio eradication. Relative hazards are classified according to risk groups 1 to 4. Risk group 1 represents the lowest threat to the lab worker and the community, and risk group 4 the highest. When the biosafety levels increase, the requirements become more stringent, as shown on screen. As a result of near-global immunization through OPV and IPV, wild polio viruses are classified at a minimum biosafety level 2. This is recommended for all countries. To guarantee safe handling of the polio virus and infectious materials as eradication nears, specific practices as shown in box 10 must be implemented. The number of unimmunized, hence vulnerable people in the world will grow once immunization stops. Consequently, any possible transmission of wild polio virus from the lab to the community will become a global health threat. The biosafety requirement would therefore increase from BSL-2 to BSL-3 to reduce the risk of virus circulation in an unimmunized population.
Thank you, Dr. Vaccine, and thank you for watching. Tune in for our next episode. Bye.
Bye.
Bye. See you next time.